I think you've probably heard the answer to this already, so I'll be very brief. Will we need novel combinations to cure hepatitis B? The answer is seemingly, and you've seen the platform set by the previous speakers, is almost certainly yes. The problem is it's proving quite difficult, and the area is quite fraught, even though the science at present is excellent. So those are my disclosures. We have very limited clinical data to date, looking at clinical trials of combination therapy, but what we've seen is encouraging but clearly not sufficient. But there are several steps in the replication cycle of hepatitis B that are clearly druggable targets, and as the science improves, we may safely be able to also target host cellular processes that are involved in HBsAg production, for example, cccDNA, and these might provide another avenue as well. But the safety of combination therapies is going to be paramount because we have a relatively safe long-term maintenance therapy with nucleoside analogues. There are lots of strategies. I won't elaborate on all of the different targets that are available. You've heard elegant presentations, but clearly these multiple strategies lend itself to the concept that we can combine these druggable targets and immunotherapies to find a way forward for hepatitis B cure and functional loss of HBsAg. But there are several difficulties, and the virology of hepatitis B poses enormous difficulties for cure. For one thing, this is a virus that, as you know, integrates into the host genome, making this a relatively inaccessible cellular target. The stable episomal cccDNA, as you heard, acts as a template for transcription of hepatitis B messenger RNAs, but HBsAg can be transcribed both from integrated cellular HBV genomes as well as cccDNA, making it very difficult, in fact, to conceive of eradication of HBsAg where the predominant source of the HBsAg might, in fact, be from integrated viral genomes. And that would specify differences between the E antigen positive and the E negative patients because although we can't quantify this precisely, probably much of the HBsAg in long-standing E antigen negative, anti-E positive patients with hepatitis B is in fact derived from integrated viral genomes. This excess production, as you heard elegantly from Massimo, and the release of subviral particles obfuscates or is blinding the immune system, rendering a unique system of tolerance which will be very difficult to overcome. And even though we have new drugs, there's always the prospect that combinations are going to be required because of the prospect of resistance, for example, with the capsid assembly modulators. This is a clever little virus, but at the same time, there are many steps in the entry, the release of the virus, the production of cccDNA, incorporation into integrated viral genomes, viral assembly, packaging, HBsAg production that lends itself perhaps to targeting with combinations. So there are exploitable targets, if you like, which you've heard. There's also the possibility, which you've heard, of possibly trying to overcome the dysfunctional T and B and natural killer cell response, which again might lend itself to combination therapy. And here the combination seems obvious, that a reduction in viral replication and a reduction in HBsAg would be probably a first step in restoration of an improved immune response. So what evidence do we have for combinations to date? I think much of the evidence started with perhaps hepatitis D infection. I won't have time to go into all of the details, but we've seen already that the combination of pegylated interferon and mercludex, which, as you know, is a peptide which binds to the entry receptor of hepatitis B, seems to result in more profound reductions of hepatitis D RNA than peg interferon alone. So that's a very promising avenue for treatment of, for example, Delta. And various dose-finding studies have been done with 10, 2 milligrams, etc. And again, you can see the combination results in quite a profound reduction in HDV RNA with patients, a high proportion, developing undetectable. Not shown on the slide, but shown in the data is the fact that even though this combination is effective at reducing HBV RNA levels in blood, it does not result in meaningful loss of HBsAg. And I'm sure, as you'll hear later, probably cure of Delta, certainly going to need combinations, but will presage the need to, in fact, cure two viruses and result in loss of HBsAg to stop the spreading pathway of hepatitis D virus infection. Nonetheless, this is an example of the direction of travel of combinations. I won't have time to show you the Replicor and the lonafarnib studies, but again, these are combination studies in Delta. So what about combinations with capsid assembly modulators? Here it's interesting, and you've seen this slide before. What I want to point out is the fact that these are potent capsid assembly modulators, and we have evidence, as was pointed out, of target engagement with a profound reduction in hepatitis B DNA and RNA. I'd like to draw your attention to this study, which is study 202, recently presented. This involved a carryover, but involved e-antigen positive patients, and it may be important to note that they were e-antigen positive patients who were either treated with entecavir and the Assembly Biosciences HO731, 731 CAM, or they were treated with just entecavir plus placebo. And as you saw earlier, there is a much more profound decline in hepatitis B RNA and a more profound decline in hepatitis B DNA when the combination of the nucleoside analog entecavir was used in the E-positive patients versus just entecavir alone. And that was statistically significant. So that's very encouraging. It has been suggested, in fact, that there might be an early signal that there is a reduction in the pool of cccDNA. I think this needs confirmation, and a very preliminary suggestion that there was a minor drop in HBsAg concentrations. Remembering these are E-positive patients, and perhaps much of the HBsAg is transcribed from episomal cccDNA rather than integrated viral DNA, and that perhaps points to how we are going to have to use different combinations in different categories of patients. Nonetheless, here the combination was clearly useful. And the usefulness lies in the fact that although nucleoside analogs will inhibit nascent minor strand and plus strand DNA synthesis, they really have no meaningful effect over the short and medium term on concentrations of cccDNA. But by giving a capsid assembly modulator, one is almost certainly affecting the binding of the pregenomic RNA in the capsid, and this is evidenced with a reduction in pregenomic RNA measured in serum. So we have clear evidence of this. We're not quite sure whether this is affecting the replenishment pathway of cccDNA, whether it's infecting the de novo entry pathway, resulting in the production of cccDNA. That's unclear. And we certainly have no profound evidence that it would affect transcription of HBsAg from integrated viral genomes. But nonetheless, it's an attractive concept, and we can clearly see that we are reducing transcription of the messenger RNAs from cccDNA. Again, combinations of RNA therapeutics. You've seen the slide before. We're developing very potent agents. They clearly are targeting HBsAg that is derived from both cccDNA as well as from integrated viral DNA. We've learned some lessons. And again, this may be an important first step in a combination that aims to reduce the HBsAg concentrations so that less antigen is presented to an exhausted T and B cell immune system. And it might be a first step in hopefully ensuring that any immunomodulatory therapy we attempt to apply, whether that is adjuvated vaccines, toll-like receptor agonists, etc., as you heard, would be more efficacious in the presence of a reduction, and these can be reductions of 2 or 3 log in a very short space of time in HBsAg concentration. The proof of this is lacking at the moment, but it should be an area where we are able to derive evidence very quickly with combination clinical trials. So this preliminary data involved a combination of an RNAi, JN3989, a CPAM, capsid assembly modulator, JN6379, in patients already on nucleoside analogs. It only involved 12 patients who were given concurrent doses, but you can see the reduction in HBsAg and the log reduction in HBsAg. This is promising, and phase 2 clinical trials with the triple combination have begun. Antisense oligonucleotides, although the action may be different, the principle is the same, and we would hope that with potent second-generation compounds with better stable delivery into the hepatocyte, we would in fact see the profound reductions in HBsAg that might be the important, necessary, and critical first step that would allow immunomodulatory therapies to be more efficacious. So we are seeing with various compounds dose decline concentrations in HBsAg that we can build on. It was pointed out that the toll-like receptor agonists have not been effective, although they induce an immune response. We've not seen reductions in HBsAg, so again, there's critical missing information as to why that's not occurring. But we do know that these are oral compounds which are potent, particularly the second-generation compounds, inducers of interferon. We have markers that would assist us in verifying this process. And if these compounds are deemed to be safe, again, they will go into clinical trials in combination with nucleoside analog suppressed patients. And I'm going to keep my talk brief because I think you've heard much of what we heard and we are running late. There are very elegant models in experimental systems, as was pointed out by Massimo. And the data from Protzker's lab, in fact, serves as an elegant model of how the appropriate sequencing of combination therapy, at least in an experimental model, is effective. So that when a small interfering RNA was given to these animals, there was a reduction in HBsAg. If this was followed by an adjuvanted therapeutic vaccine, the animals became HBsAg negative, suggesting that there's a very simple sequence, knock down HBsAg and improve the immune response. Of course, there are major caveats. We're not at all sure that this mouse experimental model in any way approximates human tolerance after long-standing hepatitis B virus infection acquired in the neonatal period, but nonetheless, it does provide perhaps some inkling of a sequence that could be applied in humans to follow. So my conclusion is, well, we need novel combinations. We almost certainly are going to need synergistic mechanisms to result in a decrease in HBV transcription, loss of cccDNA, perhaps altered epigenetic regulation and immune modulation, or immunologically stimulated, safe, controlled hepatocyte cell turnover. We are at a point where nucleoside analog suppressed patients are being included in many clinical trials. This should aid recruitment into clinical trials because we have many of those patients. And trials, as you saw, are progressing with combination therapy as additive and synergistic effects are being sought. Most importantly, I think the trials and the development of these new agents is going to provide us with some very interesting insights into hepatitis B. We're going to learn a lot more about how this virus replicates, how HBsAg is produced, how it's chaperoned through the cell, how we can block its assembly, how we can improve its degradation. And that's where the real advantages will come in the next 4 or 5 years that might make realistic propositions for combination cures possible. Thank you very much.